Okay, so today we are going to complete chapter 13, which is lipids continued. Okay, so last time we talked a little bit about the lipid bilayer, and I said what we would start with at this time. So it shows you, you can see it's much better. You have your hydrophilic, the water-loving portion, which is good for your extracellular fluid, and then you have your other layer down here, which is also hydrophilic, and it's good because your cytoplasm is mainly aqueous, so it is water loving. But then you can see everything else, it is all super chaotic. So we see carbohydrates, we see glycolipids, cholesterol, glycoprotein. So these are all words we're going to see. Phospholipid molecule, phospholipid bilayer. So it's going to come back. So we'll see these terms later, okay? All right, so the lipids found in cell membranes can be categorized into various ways. The phospholipid is the first way, and this is a lipid containing phosphorus. Glycolipid, something that contains a sugar, a lipid that contains a sugar, and then the sphingolipid that contains an unsaturated amino alcohol, sphingosine. Okay, so they're classified based on what they contain. So if we look up here, we had glycolipid. This is something that contains a sugar. Okay, phospholipid contains a protein. All right, so these are some other types of structural differences that are important. These are the structural components of some important membrane lipids. So we have the phosphoglyceride, which is also called a phospholipid, is a phospholipid. Then we have the sphingomyelin, which is a phospholipid and a sphingolipid. And then we have the cerebroside, which is a sphingolipid and a glycolipid. So we have three subtypes, okay? And we can see here we have the alcohol, amino alcohol, sugar, hence of the glyco, phosphates in these two, which is what makes them phospholipids. And then we have a glycerol. These two have a sphingosine group, which is what makes them classified as such. And then they all have at least one fatty acid group that's in common. All right, so phosphoglycerides and glyceryl phospholipids are the most abundant phospholipids in the cell membrane. Okay, so if we look back up here, that's this kind right here. There are two types of them that are common. The one that contains the ethanolamine as the amino alcohol, and the one that's going to contain, I should say, choline. Choline as the alcohol. Okay? And so up here, this is an example of that, and you can see that the ethylamine or the choline are the differences in that amino alcohol group. Okay. So two different subgroups based on that. Okay, so if we're trying to keep track of our overall flow chart, from the last time, let's see if we can pull that up. Perfect. Okay, so we are looking at sphingolipids, and we have the sphingomyelins and the glycolipids. But then we are in this phosphoglycerides when we're talking about the two groups on the last slide. Okay, so we were under that subgroup. So now, what is an emulsion? Emulsion is a dispersion of two liquids that do not normally mix, and so sometimes it ends up making kind of a cloudy suspension. And so if you think about maybe vinaigrette dressing, the oil and the vinaigrette, when you shake them up, they look real cloudy. And they will separate out over time, but when you first mix them up, they're considered to be kind of like an emulsion. Other examples would be things like milk, things like blood. Those are other examples of emulsions. All right, so what is a sphingomyelin? So this is our other group if we go back to here. So we're looking at this group right here now. So what are they? So a sphingomyelin is a sphingolipid that contains a fatty acid unit, a phosphoric acid unit, a sphingosine unit, and a choline unit. So let's look at this structure. I think it might be down on the next one. Yeah, it's on the next one, right here. So it has the sphingosine, the phosphoric acid, the fatty acid, and the choline. Okay. So most animal cells are going to contain sphingolipids, specifically called cerebrosides. And this is a specific type of sphingolipid that contains the fatty acid unit, the sphingosine unit, and a galactose or a glucose. So remember, this is the one that contains a sugar. The ganglioside is the sphingolipid that contains a fatty acid unit, a sphingosine unit, and a complex oligosaccharide. So oligo usually means few, so it's more than 2 but less than like tens of thousands. And so we call it oligo because it means few. So the best thing to do for this chapter is to make, I would say, flashcards to help organize the different ones and what they're made up of. And that's what you need to know. Structurally, how are they different? And Sapling's going to give you lots of opportunities to identify based on structural differences. Okay, so looking at this one, this is the sphingosine group right here. That's common to all sphingolipids, so it's what makes them unique. And then the sphingomyelin down here, also known as a phospholipid, because it has the phosphoric acid. Okay, so it has the sphingosine and the phosphoric acid, which kind of makes it a little bit of a hybrid, a 2-for-1, because it has both those features in it. So what does a cerebroside look like? So it's still going to have the sphingosine unit and a fatty acid unit, but instead of the phosphoric acid and the choline, it has a sugar. Remember, and the sugar is either going to be galactose or glucose. All right, so membrane proteins. So if we go back up here to the top and look at our lipid bilayer, we have in here, we saw peripheral and integral proteins, okay? Surface proteins, and then there's the globular proteins, okay? And then the protein transport, which we won't see quite yet. So there are 2 major classes of proteins in the cell membrane, the integral and the peripheral. The integral is one that spans the entire lipid bilayer, meaning it's going to go from one side to the other. Okay, so the protein is going to be quite large. The peripheral one is one that's going to be loosely associated with the membrane surface. So if you think about peripheral vision, that's your side vision, right, what you can see off to the side. So let's go back up and look at integral and peripheral. Okay, so peripheral, it's here on the side, whereas the integral, it's integrated throughout the entire lipid bilayer. Okay. So what's the last one on here? Carrier proteins. These are things that are going to facilitate the passage of certain molecules, such as hormones and neurotransmitters. And they do that through specific interactions between the protein and whatever molecule is being transferred. So that's the protein that we saw in the very end that was called a carrier or transport protein. All right, so let's practice classifying these lipids as either a phospho, glyco, or sphingolipid, and then some lipids we can say can be given more than one classification. So this is going to be your in-class work. Today should be the 13th. Sorry, I'm having to look at my calendar. In-class work. And we're going to answer 2A and 2B. Okay, so looking at the first one, the first thing I see is that there is a glucose right here. Okay, and I'm sure y'all heard that. We're watching Dr. Pohl. He's watching Dr. Pohl. I am trying to do the lecture. Okay, so this presence of the sugar, the glucose, oh, I keep forgetting that color doesn't show up well. The glucose classifies this one as a glycolipid. Okay, so hopefully you can see that on there. Now the other ones we have to look for are phospholipid, and I don't see any phosphorus on here on the first one, so I'm going to say it's not a phosphorus. But we just briefly looked at the sphingosine unit, so maybe we need to go back and look more specifically at what that looks like. Okay, so looking at the sphingosine, it's going to have 3 carbon backbone, maybe a nitrogen, alcohols, and then this long group right here. So now that we've seen it, let's go back and look. And I do see it. So this right here is my sphingosine. So in addition, I'm going to classify this as, and this is a really really strange word to say, sphingosine. So this is also going to be a sphingolipid. Okay. All right, the next one. So the next one, let's see, we're going to start off. And if I look here, there is also a glucose unit. It just is written the other way. So we're going to say that this is a sugar, so this is a glyco. And then I also see here that we have this phosphate group. So that's definitely a phospho. But I don't see anything that looks like the sphingosine. Okay, so then I would say in this case, this one is only going to be classified as a phospholipid. Okay, and looking at this more closely, this is not glucose. I'm not sure what kind of group it is, but glucose would have the CH2OH on it, and it doesn't have that anywhere on here. So while it is a 6-membered ring, there's no oxygen integrated inside the ring, so it is not a glycolipid. It would only be the phospholipid because the glucose group is not there. Okay, so that's a good example of how to do that wrong, because now I can see that the glucose unit is not there. Okay, so don't get rid of your in-class worksheet quite yet. We're going to have one more at the very end of this chapter. Okay, so the last group we're going to talk about are our steroids. Okay, so this was, if we think back to our organization, steroids were, so we've done this one, gangliosides, glycolipids. The steroids was this last group right here that we were going to save for very last, and here we are. So steroids are a little bit different because it's a lipid that has a 4-fused ring structure. Okay, so what that looks like is this: 4 rings. And by fused, it means they're connected like this. Instead of being independent rings, they are fused together. And so this is considered to be like the backbone. Okay, and everything else is going to be built off the backbone. This is kind of like your base model car, and anything extra is considered to be extra. All right, so cholesterol. Okay, we often hear about cholesterol as being kind of a bad word when it comes to your overall health. It does not occur in plants, but it is the most abundant steroid in the human body. It is a precursor in the synthesis of sex hormones, adrenal hormones, and vitamin D. So cholesterol is a necessity, even if we don't think about it as being a necessity because it has such a negative reputation. We have to have it in order to have all these other things our body needs. Now, excess cholesterol that's not metabolized by the body is released from the liver and transported by the blood to the gallbladder. And that's where we get into trouble, because you're loading your body up with cholesterol that it doesn't know what to do with besides to try to get rid of. So cholesterol is good, but too much cholesterol is not good. Okay. So there are a couple of things we're going to talk about. One is the steroid hormones. But before we do that, I want to talk about cholesterol and its purpose in the membrane. So let's go back up here. So you might have seen that there was cholesterol in the membrane when we saw this example. There's actually quite a few little cholesterols in here. So cholesterol is essential because the membrane needs it in order to stay fluid, but too much cholesterol in the membrane makes it too fluid, and it ruins the integrity of the bilipid layer. So while you want some cholesterol to have fluidity in your membrane so it's not rigid, you don't want so much cholesterol that the membrane is too fluid and is not able to protect the things it's supposed to protect, which is the cytoplasm and everything that's inside of that cell. Okay, so kind of a weird side note, but it does show up on sapling occasionally about what cholesterol does, and sometimes it shows up as a multiple choice question on exams. All right, so let's talk about hormones. So hormones are the chemical messengers that are released in one tissue, transported through the circulatory system to one or more tissues. One group of hormones is known as the steroid hormones because they are synthesized from cholesterol, which is also a steroid. The two main groups of steroid hormones are the adrenal cortical hormones, such as aldosterone and cortisol, which are produced by the adrenal gland, which is located adjacent to the kidney. Now, the sex hormones are a class of steroid hormones secreted by the gonads, which are the ovaries or the testes, the placenta, and the adrenal glands. Okay, so these are both really important groups of hormones. They all do very different things, but they're important for mood regulation, thyroid function, for women, your cycle, your menses, and all of that. And so they're very important, and cholesterol is how all of those hormones are built. So it is important. Now, bile salts are part of this whole thing because bile salts are also considered to be derived from cholesterol. They have that 4-ring fused backbone. Now, bile is the yellowish green liquid that's produced in the liver. And the most important constituents of the bile are the bile salts themselves. So anytime, I have always suffered from migraines, and so when I get sick, one of the things that sometimes I'll end up doing is getting really nauseous and throw up. And I normally throw up stomach bile, which is really bitter. It's very acidic. It doesn't taste good at all. So if you've ever thrown up something like that, it's most likely your bile, your stomach bile, and it's that yellowish green liquid. All right, so what are some of the important steroid hormones? We talked about aldosterone, which regulates salt metabolism, stimulates your kidneys to retain sodium and excrete potassium, so it helps maintain that electrolytic balance between sodium and potassium. Cortisol, which is hydrocortisone, stimulates the conversion of proteins to carbohydrates, which is an important part of energy metabolism. Progesterone, which regulates the menstrual cycle, maintains pregnancy. Estradiol, which is an estrogen, stimulates female sex characteristics, regulates changes during the menstrual cycle. And then testosterone, which is an androgen, stimulates and maintains male sex characteristics. So, pretty important group of steroid hormones. So what are bile salts? These are examples. The cholic acid and the sodium glycolate are both bile, well, one is an acid and one is the bile salt. But you can still see the 4 rings, very similar to what we saw in the very beginning. Okay, so they all share that in common. All right. So last question, we're going to answer number 2 and number 3. Oh, I don't know if I left myself enough space, but hopefully I did. All right, so it says, which of these compounds are steroids? Vitamin D, cholic acid, or lectin? So all steroids are non-saponifiable lipids, and the only ones here that are steroids are vitamin D and the cholic acid, which we had already seen that one on the previous slide. But vitamin D is another important compound that's going to have the 4-ring structure. Okay. All right, so draw the basic steroid skeleton and label each ring with the appropriate letter designation. So Sapling has you practice this. Okay, so we're going to draw hexagons, 2 hexagons side by side. Then we're going to draw one like that. And then the last one is drawing a pentagon, or we can make it look like home plate, if you've ever played baseball or softball. And then we label them A, B, C, and D from left to right. Okay, so Sapling's going to have you draw that, and that's the main reason why I'm having you look at these now, is so that you've seen it before. I didn't time the lecture, so I'm honestly not sure how long it has gone over, but hopefully there is plenty of time if you would like to work on Chapter 17 homework, to ask questions. This is a great time to do so. Hopefully the questions that are left in the homework are ones that you can easily work through, but Dr. Morris or Dr. Spencer will be able to help you with that if you have any questions. I hope all is going well in class. If you have any questions or concerns, you can always email me, and I can try to help you as soon as I can. Let's see, at this point, we have 4 lectures left, chapter 14 and 15, and then your exam, and then the final that will be on Wednesday, December 4th, at 10:30 to 12:20.